For AP Biology or College Biology, we're going to be covering Unit 3, which is all about cellular energetics. Specifically, we're going to get into a brief overview about the enzyme in general and its structure before actually diving into the more details of enzymes. But before we get started, let's make a note of the disclaimer listed below. All right, now that we've covered that, let's get started. So when we're talking about the enzyme structure overall, it's a very complicated structure when we look at it in general. It comes in different forms, different shapes, different sizes. But what happens is enzymes are connected to energy. There is a relationship between energy and enzymes. What happens is I want you to think of someone that is a runner and they need to go ahead and run up a hill. And if you run up a hill, you might think to yourself, let's imagine myself running up a hill. When you run up a hill, you exert a lot of energy to go up that hill. It takes a lot of work and effort to just climb up that hill. Well, what if I told you that you got these special sneakers? And those special sneakers give you an extra boost to get you from the bottom to the top. So that special boost is what an enzyme is. The thing is, we need energy for a lot of things in our body, such as breathing, or in plants, what is called photosynthesis. Basically, cellular respiration, which we use as humans, and photosynthesis are two processes that either consume or use up energy or release energy. So we're going to talk about later on this topic later down the line in the unit. But for now, you just need to know that breathing takes energy at some point and photosynthesis utilizes energy as well, just in different ways. It either comes out or comes in, but you don't need to know that for now. You just need to know that there is energy in general, just like a person running up the hill. However, what enzymes do is they basically are that energy boost, like those sneakers, to get up that hill. They provide the extra boost that is needed to get from the start to the finish line. They basically are like our battery pack for us to still get to the same place, but with a little bit of extra push. Enzymes are called catalysts, and that is sort of what we mean by those extra shoes, those special shoes that basically help accelerate or get us to the very end. We still get there from the beginning as a runner to the very end, whether or not we have that enzyme or that energy boost or that extra pair of shoes. However, enzymes just work to give us the push to get there just a little bit easier. And enzymes in general are used for things like metabolism or replication or gene regulation. These are all topics that are covered later on for Unit 3 or just AP biology in general. But for now, you just need to know that enzymes work as catalysts, which are like those sneakers that give you the extra boost. Okay, so let's talk about a recap of Unit 1 to really understand where enzymes come from. If you remember, in Unit 1, we talked about amino acids and we talked about macromolecules. And if you remember, amino acids form eventually polypeptide chains. So this one amino acid will come together with other amino acids and they form what's called a polypeptide chain. And oftentimes, one of those amino acids could turn out to become enzymes. So that's where they fall together. Just a recap of understanding what enzymes fall under in terms of category. Now, when we talk about enzymes, we can talk about how they get to where they are again by different structural levels, they're called. What I created here is a little table, and this is going to help you understand the four structures of how enzymes become to be. And you do need to know this and the differences for the AP biology exam or even any college biology exam. The first structure is the primary structure. And sometimes it's written down with a little one and a little degree. So I just want you to note that symbol is the same thing as saying primary or primary structure. And what that is, it's a bunch of amino acids that formulate a sequence. And they're seen as this like little bead-like structure here, and it's really just connections of amino acids that are connected together. Now, if you have a secondary structure, which you can think of 2D, or like think of that there has to be some sort of 2D structure, what happens is they could form either alpha helixes or beta sheets. So what alpha helix is it sort of as a twisty turn shape, and beta sheets are these like long-winded shapes that formulate. And I have these pictures here so you could see there is not a flat structure, but there is some folding that occurs. So the primary structure can fold on itself or fold in a certain way that can be either alpha or beta, meaning sheets or helixes. So alpha helixes or beta sheets. So that's a secondary structure. And you are expected to know this for the AP biology exam. And you need to understand that if they tell you it's alpha helixes or beta sheets, they're really enforcing the idea that they're talking about that secondary structure. Okay, so now let's talk about if there's some sort of depth. I want you to think of a 3D printer or a 3D structure of any way, and that's what tertiary is. Tertiary structure. If you think of tertiary, it means 3, 2, and meaning three, the number three. And when we think of tertiary structure, we're thinking about that 3D folding. So now we have a bunch of foldings together and they come out of the page. If we could protrude this out of the page, that's what a 3D structure would be. That is what a tertiary structure would be. And lastly, when we're talking about a 4D shape, we're talking about a bunch of these together to formulate a multiple polypeptide unit. So it's a bunch of them together. So basically, when we're going from primary to quaternary, we're getting bigger and bigger and bigger each time. So that's important to note when we're talking about the shape of enzymes. Please note that the enzyme fully is in structure when you get into that tertiary shape. So I just want to point that out. So if they ever ask a question about some sort of enzyme and how do you know, is it when it's primary, secondary, tertiary, or quaternary, we start to see the development of that enzyme when it starts hitting that tertiary structure. Okay, now let's take a look at enzymes more in depth. And there are two different theories or two different ways in which enzymes were previously stated as working. And the first one is called a lock and key, which we don't accept as true anymore. And now what we accept is what's called the induced fit model. The induced fit model is the model that we use to really talk about how enzymes really work. And we're going to go more in detail in the next slide about what an active site is and what a substrate is. But for now, you just need to know a lock and key was the old version, and the new version is called the induced fit. Okay, now let's cover what the induced fit really means. So I want you to take a look at this picture on the left-hand side here, and I want to focus on this picture right here. The thing is, remember we talked about that booster pack. Your body or any sort of reaction in your body will have certain enzymes. And so that's what this structure is right here. However, enzymes are not going to work unless they are turned on, essentially. And the way they turn on is because they're going to have some sort of substrate. That's this structure right here, which goes and attaches to the enzyme itself. It basically turns it on. And so that's where the lock and key method originally came in, was the fact that you could essentially have these enzymes that would connect to the substrate at a specific point, that specific point being the active site, and it essentially will turn on the enzyme when it becomes the enzyme substrate complex to then work properly. But now we don't think it's just a lock and key where it just fits exactly the same way. We've now accepted the new mechanism, which is called the induced fit mechanism or induced fit model, which basically states that you could have what is called a substrate, and the substrate goes and looks for an enzyme that it needs to attach to make sure that it can attach to what's called the active site. The active site is the location in which both bind together. Once both bind together and it fits properly, and I want to point that out, what fits properly, which I'll explain in just a second, it produces what's called the enzyme substrate complex. Because once it fits properly, the substrate attaches to the active site to the enzyme. Essentially, the enzyme turns on and will now produce the proper products that are necessary for the enzyme to have its purpose. So once it's attached, the substrate becomes essentially the products that it needs to be and is released. Now, if you remember, I said that it needs to basically conform or become a certain shape. It needs to basically envelop into the shape. And so that's a lot of words to think about. What we're basically saying is we have this enzyme when it becomes the enzyme substrate complex. And in the induced fit model, what it says is the enzyme will essentially change its shape to better bind to the substrate. The thing is, with the lock and key model, if you think about a lock on a door or a lock on maybe like a suitcase or something, that lock is always going to remain that shape, and that suitcase hole is going to remain the same shape. We basically say that it's one lock for every one key. However, for enzymes, this isn't the case. The induced fit model basically says that there's enzymes that exist and that will alter its shape based on the substrate to bind together. So it induces. So that's why it's called the induced fit model. The active site, or the location in which it binds, is going to conform or basically alter its shape to better fit for that substrate. And basically, once the shape is finally formed, we call it stabilized. We understand that during this step right here, we have what's called catalyst lysis, or it becomes a catalyst. And so enzyme and the substrate formulate together to form this catalyst to produce the eventual products. The thing is, which you will learn later on down the line, enzymes can have many, many different substrates. There is no one key to one enzyme, but you could have one enzyme for basically millions of different substrates. But the way that it induces or fits properly is going to allow for efficiency to produce the proper products in the proper form that it needs to be. So that's what the induced fit mechanism basically states. It says we have some substrate, we have some enzyme, they attach together at the active site. However, the enzyme will switch the way that it fits around the substrate to eventually formulate the enzyme substrate complex to eventually produce the proper products that are necessary for its processes. So that's the induced fit mechanism. Okay, so now we've talked about that. We're going to only cover this briefly, but you're going to cover this later on. There are different ways in which the enzyme can work, and one of them is called allosteric regulation. And the thing is, you can have an enzyme and you can have an active site. So let's say that this little location right here, and you can have a substrate as shown here. However, what happens is there could be another location on the enzyme called the allosteric site, in which you could have a substrate copycatter, I want to say, that sort of binds to that enzyme and alters the functionality of that enzyme to therefore no longer attach the substrate that originally was intended for. So essentially, what you have is you have this enzyme that has a different key that can attach to it, which causes the original substrate to feel like not wanted. And so, what ends up happening is the enzyme will change the purpose based on what really attaches to it. So, you could have different things called competitive and non competitive inhibitors, which we'll cover in a different section, but for now, you just need to know that guess what? If you have an enzyme, you could have a substrate or you could have some other item that can come in and attach to it, which can alter the functionality of that enzyme. And these are known as cofactors sometimes, or there could be environmental factors that could alter the way that the substrate and the enzyme work together. And this also can alter the way the enzyme produces some sort of catalyst-based reaction. So I just want to put that out there that there is factors such as temperature, such as pH that we talked about in Unit 1 that alter the way the enzyme actually works. And so they can be effectively changed by these factors and or the ability of some sort of different solution that could be a different substrate, or I should say an inhibitor or allosteric site that could alter the ability of that enzyme to work that way it needs to be. And that will be covered in a later subsection as well for Unit 3. So if you weren't able to really grasp that, that's okay. We're going to cover it in a later unit. Now, I want to take a moment to try to start this quiz question. So take a moment, freeze your video, and read the following question. After you're completed reading the question and potentially coming up with an answer, we'll review the question together. Okay, now that you're done reading the question, let's review this together. The question basically says a chef is preparing dough for bread. According to the induced fit model of the enzyme activity, how does the enzyme in yeast, a typical protein, interact with the sugar in the dough to ensure proper fermentation? This question, based on your knowledge, may seem out of scope because they talk about fermentation, which we'll cover later on in the unit, but it really isn't because you're talking about the relationship between the enzyme and the sugar. So basically, when you read your answer choices, let's try to review them together. So the first one says the enzyme in yeast is rigid and only works if the sugar molecules perfectly fit into it. So here's the thing with most, if not all, science questions, we want to stay clear of extreme options because in science or STEM questions in general, extreme answers are not really favored. So words like only makes it seem like an enzyme only has one functionality. And we also mentioned that it's rigid, which we now know is not the case. Remember, you could have different substrates that come in, you can have inhibitors or whatever they may be that alters the way the enzyme works. It isn't a lock and key method, therefore it is not rigid. And so that answer is not favored. Let's take a look at answer choice B, which states that the enzyme in yeast is slightly changes the shape of the sugar to better fit into the enzyme and ensuring fermentation. This seems like a good answer. However, I'm kind of skeptical about this because we're saying that the substrate or the sugar is going to change its shape. However, based on the information that we talked about, remember the enzyme is the one that's going to change the shape in a way to fit that substrate. So I'm going to keep this as a maybe for now and see if there's a better answer choice. Please note for AP biology or most, if not all, standardized tests, you're not really finding the correct answer 100%. You're finding what's the best answer for that question. And I want to keep that in your mind because if you're reviewing questions and you see something like answer choice B and they don't have a better answer, then that answer would be favored in that case. So let's go to answer choice C, which basically states the enzyme in yeast adjusts shape to better fit the sugar molecule, helping break them down and speeding up the fermentation process. I like this answer better, not because it talks about speeding up or breaking them down. It just makes it very much known that the enzyme in the yeast is adjusting its shape. So compared to answer choice B, I like that answer better because we're making note that the enzyme is going to change its shape. And answer choice D basically says the enzyme in the yeast permanently changes its shape after binding to sugar, allowing it to catalyze fermentation only once. Once again, these are extreme answers. It doesn't give exceptions, so I really don't like permanently either because that's an extreme answer. So I favor the answer choice C, and C is the correct answer. So I hope you like this brief debriefing of what enzymes really are in terms of structure. We hope to create more videos like this with practice questions in the future. Make sure you hit that subscribe button and make sure to like the video, and we hope to see you back soon. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams. We can't wait to help you succeed.